Hello, my name is Dennis Province from the National Resource for Quantitative Proteomics. And the purpose of this video is to give you an overview of the national resource, as well as to prepare you for an initial consultation if you decide you want to use the core. Some of the topics in this video are going to be the core workflow and timeline, some questions about your work, some experimental options that we offer at the core, sample preparation advice, and a word about final reporting. This video is geared for people that have never done an omics experiment before, so that when you use the core, you'll be able to get the most out of our services. Principal investigators from across the country, including you, usually contact the core by email to set up an initial consultation. This consultation is now done by video conferencing, where we communicate with you, the principal investigator, about your experimental goals, explain some of the experimental design choices that you have, and talk about your sample types. If you decide to use the core, then you would need to send us your samples for processing. The following steps are all done at the core. We can do sample preparation, which includes protein quantification, cell lysis, desalting, some limited detergent removal, as well as enzymatically digest your samples. And if needed, we can do isotopic molecular tagging or enrichment. It's important to know that some labs choose to do their own sample preparation. And you would need to ask before you process your samples, because once you have processed your samples, if they're done incorrectly, then the data analysis will be poor and you won't be happy with your results. It's always best to ask before you start your experiment so that we can give you advice on how best proteomics can be done. Or you can just send us the cell pellets or tissue and let us do the processing. The purpose of sample preparation is to take your proteins, separate them from the rest of your samples, and then enzymatically break those down into peptides. Those peptides are then analyzed by mass spectrometry. Chromatography is used to separate these peptides, where peptide masses and the masses of the fragment ions can be measured, as well as the retention time. And this information can be searched against a library to get protein identification in your control groups and in your treatment groups. This information is then fed to our bioinformatics team that then prepares your final report. This final report will be communicated back to you by another video conference about 6 weeks after we receive your samples. So what exactly are your goals? Most people that use our services do discovery-based proteomics. This is a broad survey of the most abundant proteins. With good sample preparation, we have found in the core to be able to identify in mammalian cells 6,000 to 7,000 proteins, or 5,000 to 6,000 proteins identified in mammalian tissue, and between 500 and 750 in a serum or plasma run. If you're doing a targeted approach to proteomics, that means that you tell us the small handful of proteins that you are interested in monitoring its concentration. And we can do that analysis for you as well. Questions that you need to be thinking about before you contact the core is the number of samples that you want to run and the number of different types of groups. Are you going to have gender differences or batch differences? Are you going to have a control group and different treatment groups? How do you want to compare these? This is all important to know up front before we do the data analysis. Also, we're going to be interested in the types of samples that you're running. Are these tissue samples, cell cultures, or serum? And what organism are they from? Our grant is through the NIH, and we need to know when you use this data to publish a paper or to write a grant for our reporting. The goal of your experiment is to give you a list of protein IDs for your control groups and your treatment groups and the log fold changes that occur in different proteins because of your treatment. Some of the experimental options that we have here at the core is to do a labeled experiment, which is sometimes called a TMT or a tandem mass tag. This experiment is limited to about 20 samples. Another type of analysis is called data-independent analysis. This has become more popular, especially for larger sample groups. It's less cost per sample and there's no labeling involved. Have you thought about the replicate samples? In proteomics, statistical analysis is a must. It is suggested that at the minimum, you have at least 3 replicates for cell cultures and 5 replicates for tissue samples. Now some advice about sample preparation. You need to send in each sample about 100 micrograms of protein to be able to do both the DIA or the TMT experiments. If you're thinking about an enrichment experiment, then you need to send double that. And if you're doing working with plasma or serum, 100 microliters works fine. For cell cultures, 100 micrograms equates to about 10 million cells. It's important that you avoid all non-ionic detergents in your sample preparation. And it is suggested that you do a protein quantification or validation, which might include a BCA assay or a Western blot. What can you expect to receive once the analysis is complete? The final report includes a second video conference where the finding of your report will be discussed, which includes tables that list all of your proteins identified for your control groups and your treatment groups. Any questions that you have will be answered, and you will be given this final report and these tables to work through more thoroughly with your own bioinformatics team. This second video conference lasts about an hour, and this report and conference is part of the service which the National Resource provides to you. It's important that you design the experiment to address the most important questions that you have and state those in the initial consultation. For instance, if you state that you want to compare treatment 1 versus the control, then when you get your final report, all the positive log fold changes will indicate that proteins have been upregulated as a result of the treatment. We thank you for listening, and if you need to get in touch with us, you can send us an email at ideaproteomics@uams.edu, or you can find us on the web at idearesourceproteomics.org. You can also follow us on Twitter at ideaproteomics. We look forward to working with you soon. Thank you.